Fire.
Turn that on.
Okay. And then is that on final?
Hit that button, and that should show up. Oh, it's starting.
We'll still wait a couple minutes.
Yeah.
Hi everyone, thanks for waiting. So I would like to introduce our next speaker, Dr. Kevin Bradley. He is a professor and state extension weed scientist at the University of Missouri. His applied research program focuses, among other things, on the development of programs for prevention and management of herbicide-resistant weeds in the areas of corn, soy, wheat, pastures, and forages. And today he will be giving his talk on Have We Gotten Lost in the Weeds? Where we've been and where we're headed with weed management in U.S. agriculture. So please help me welcome Dr. Kevin Bradley.
All right, thanks very much for the invitation, and I hope I can do that title justice. When Brent and Catherine asked me to come over, I'll be completely honest, the first response of mine was, you've got some experts already right here in Dr. Tranel and Dr. Hager that could definitely tackle this topic better than me. But anyway, I thought a lot about what the symposium was and kind of how I would tackle this issue. And so what I decided to do is I'm just going to take you through a little history of what has happened to me in the state of Missouri, and hopefully that brings us to the final, which is where because of all those things that have happened with resistance and water hemp and weed management, where I think we're headed. So hopefully it'll make some sense by the time we get there. So in August of 2003, I arrived at the campus of the University of Missouri as a brand new assistant professor, full of energy, full of life, much younger, much more hair, much thinner. And on my plate in August of 2003, on my desk was this note that said, there's a producer in Weston, Missouri. None of y'all know where that is probably, but it's north of Kansas City Airport. And he believes he has glyphosate-resistant water hemp. And I was brand new, didn't really know a whole lot about water hemp, to be honest with you. I came from eastern U.S. where there's not water hemp. And so the long story short is, it was too late to do anything that year, but we conducted field trials in 2004 and started doing research to confirm or deny whether that was true. Once I met with the farmer, found out this was kind of one of our situations that we're not so proud to tell what the history is, but this happens more often than not, and that is that it was continuous Roundup Ready soybeans, glyphosate-resistant soybeans since '96, with at least 1 to 2 applications of glyphosate per year. So as many of you already know, if you do that, you're going to create a resistant weed, and that's in fact what he did. And so we did 2 or 3 years worth of research. I was, I look back on that and thinking I was so young and so stupid and so cocky, and I had company opposition and everything else from all the industry saying you don't realize what it means to say that there's glyphosate-resistant water hemp. Don't know how I would have handled it now if it was the same situation, but anyway, we did the best we could back then, and we confirmed it eventually to be glyphosate-resistant water hemp, which was a pretty huge deal. Turns out a lot of the same was happening in Illinois about the same time or shortly thereafter, and a lot of other states as well. But everybody knows, especially you young people in here, you think it's no big deal, but I just want to take you back. If you can imagine what it was like in 2003 in weed science. A lot of you probably were barely born, but in 2003 in weed science, resistance was not anything like what it has become to be accepted today. There was some glyphosate resistant horseweed or mare's tail, whichever you prefer to call it in this state, and it's Coniza canadensis, if that makes anybody know what that is. It was in a few states. A lot of people said it's just because it spreads aerially, and that's why we have this all over. But it really, resistance, and particularly glyphosate resistance, wasn't what it is today. So that's all we were dealing with at that time. There was that one little case over there of ryegrass in California, but who cares about that? So so that's where we were. It was an entirely different feeling situation. So I'm just going to walk you through what we did, and it's not saying that we didn't make mistakes and need to do things better, because obviously we learn along the way. But what did we do after finding resistant water hemp? We did what any extension weed scientist is trained to do. We did the best we could with publications. I did training modules, I did field days galore, even went to the extreme of doing videos, which was high tech back then, man. If you could put something on a video about weeds, there was no such thing as social media and all that junk, okay? So we did all that. I went to field days. I was surprised by how many talks and how much I was talking on all of this. 5 years later, we did a survey through the state of Missouri of water hemp that was remaining at harvest in soybean fields, and this is 5 years later, and this is what we got with water hemp. And so it was somewhat disconcerting to me that all of that time and effort was spent in going to those meetings. And I'll be honest, especially during those early years, you could go to the farmer meetings and you can tell them you got to use something other than glyphosate, and you got to use pre-emergence herbicides and rotate and mix mechanisms of actions and all of that. And quite frankly, most of the response was sleep or a blank stare, and not a whole lot of adoption of what we were saying. So felt like we were beating our head against the wall. But this is what Pat spent a lot of his career on. This is what a lot of people have, and this is what makes water hemp and Palmer amaranth different. it explodes when it happens. It's not just, you know, it was found in this county on one year, and then it got outside a little bit of this county on another year. It took 5 years before it was a predominant weed problem in the state of Missouri, and still is today, even worse than that. At the time, so what was going on by then in the U.S., we had a lot more resistance. So this wasn't just Missouri. This was 2008 in the U.S. as far as what glyphosate-resistant weeds were doing. And we had evolved from this is only a mare's tail issue or Caniza issue to now we have some common ragweed. We have many more states with, oh, we got a pointer here. Many more states with water hemp in the heart of the water hemp growing area. And down south, Stanley Culpeper had been the first to find it, but it had many other states had come on board by then with glyphosate resistance in Palmer amaranth. So the situation was definitely changed by 2008 compared to 2003. By 2008, we had started something that I think has been good and worked a little better than us trying to do it alone. So around that same time, 2008, 2009, somewhere in there, the United Soybean Board finally agreed that this was a problem after several attempts on our part to write grants and everything else. And so we formed this group, and it became known as the Take Action Group eventually, but it took us a while to get that name. But since its conception, all of these individuals and agencies on the screen have come on board. That wasn't necessarily the case at the very beginning, but all the companies have come to accept it and support it, and there's some, at least 20 or so land-grant university participants across the U.S., and all housed in a format really facilitated by the USB initially, and it's kind of taken off from there. So it was a cooperative effort, and that's really what we needed. And some of you may have seen these kinds of advertisements in magazines. This was the initial concept that we have. That's why it got the name Take Action, is because our biggest emphasis area as a group of extension weed scientists sitting around a table with a bunch of marketing people, that's what we did with our money, by the way. One of the first things we did with our money that the USB funded us with is say, hey, you know what? Kevin Bradley in the state of Missouri has been beating his head against the wall and trying to do this individually and hadn't had much success. So has Aaron Hager in Illinois. So has Bill Johnson in Indiana. Maybe we need to recruit or hire somebody that knows a little bit more about marketing. So we actually did. We hired a marketing team. With all of our comments and I guess input, they boiled it down into this, and that is that growers need to take action on this issue for themselves. You know, you have to put it in their court because they're the ones dealing with it, and you have to illustrate the importance of what it is to mean to take action. So this was the big slogan, and that's why we got the name Take Action and all of that. But there was many kind of scientific slogans underneath of it. We would say something, and then the marketing people would look at us and say, that sounds scientific, we're not going to say that. We'll boil it down into something like this. I will use multiple herbicide sites of action, which of course is very, very important, but we probably didn't do as good a job as saying it. I will say back in 2003 through 2008, I was talking to farmers about PPO inhibitors and ALS inhibitors and all HPPDs and all these things, and quite frankly, that was a major mistake that I made and a lot of other people made too. That terminology, they gloss over, they don't care about that, and so we found a better way, which I'll illustrate in a minute, but that's one of the lessons learned. And so what the WSSA, the Weed Science Society in America, and a lot of other groups, this isn't just a take action thing, but what we've tried to do is stop talking about PPOs and instead talk to a grower about group 14, and that you need to rotate between group numbers and not use a group 14 over and over, or not use a group 9 over and over and all of that. So during that same time frame, 2008 all the way up really until today, our biggest, for me to go out and talk to farmers today, if I'm not asked to talk about dicamba, which I rejoice in, then the number one thing I'm talking about is mechanisms of action and resistance, and taking out these posters which we give out and walking them through mechanisms of action and how to not use the same thing over and over. And so it's really been quite good, a good dialogue between farmers. Somebody took this picture of me doing it at one of our typical farmer meetings in the winter, but this is exactly what it looks like. I'll say, Mr. Farmer, what are you planning on using? Okay, well, let's find it on this piece of paper. What is the mechanism of action in that Authority XL? You tell me, you find it. Okay, so what does that mean? You're going to use post-emergence. What are you going to use next year? And we walk through it, okay? So it's as simple as that. And that has been very, I think, rewarding, because many farmers in Missouri today come talk to me about, they don't say, it's amazing. They talk to me in numbers now, and I think it's just a victory. They come and say, Kevin, I used a group 14, and so what do you think I should use a 15 too, or should, and so I think that's just a big victory that the entire society has had. I'm going to talk about this one later, but this is my favorite one, and really where I'm going to head to at the end of my talk with where I think we're going from here. But there's numerous of slogans. I'm not going to show you all of them, but we wanted to emphasize there's things other than just herbicides, and we're going to talk about some of that. But I do think, as I talk to you about our history in Missouri, what I've seen happen probably only in the past 5 years or so is that I am starting to get through to farmers, or it's either I'm getting through to them or I'm not getting through to them and they just have it so bad that they got to do something else. I think it's probably the latter, but anyway, I like to think it's the former. But anyway, what I've seen happen in Missouri, which I never thought I would see happen in 2003, 2004, 2005, is that they're actually doing things like cover crops in moderation. I'm not saying we have every acre with cover crops. They're actually thinking about tillage in some places where it would be appropriate. They're thinking more about row spacing. They're thinking more about all kinds of crazy ideas that are happening in weed science right now about things other than herbicides, and I think that's good as well. Regardless of that, I don't think I'm going to do any more surveys, Pat. I'm tired of surveys, but this was 2014, the last one we did, and I think really what has happened in our state, and unfortunately in Illinois too, I know, and in Iowa and Kansas and everywhere that water hemp is that we're finding way more instances of multiple resistance. Our farmers may have actually adopted and done the right thing after they got glyphosate resistance, but I think it's probably too late in some cases, and I think it's also due to the spread of resistance through the pollen that water hemp can have. So for whatever the reason, we started seeing by 2014 that the problem was no longer, hey, I got glyphosate resistant water hemp. That nobody has just glyphosate resistant water hemp in Missouri anymore, I don't think. They have glyphosate and group 14 resistance and all kinds of other resistances built in. In 14, we had a population we found with 5 different types of resistances. This paper was published just recently, and it's a population we found that has 6 types of herbicide resistances all in one population. The molecular paper just came out in Pest Management Science the other day, but this was the initial dose response. So, we don't have this everywhere, by the way. There's only one population in one field that I know about, but it's just to illustrate a point, and that is Dr. Heap's website. If you're a weed scientist, you know all about Ian Heap and his website. But basically, he is the caretaker of all the information on resistance. And what he's showing here in this graph is that multiple resistance is now the biggest problem that we're dealing with in agriculture overall when it comes to weeds. We're finding water hemp pretty commonly with 4- and 5-way resistances. Like I said, we had that one with 6-way, but in Australia and other places with ryegrass, there's been some nightmares of 8, 9, 10 way resistance and all of that. So obviously it's safe to say that we need to be thinking of things other than just herbicides in my mind as I look at our history and look at what has happened. So I'll just bring you up to date. This is today as far as I know it. I think I got everything on Ian Heap's website in here. It's kind of hard to fit everything on the map anymore, but this is where we are in U.S. agriculture with just glyphosate resistance. But as I just said, there's way more than just glyphosate resistance that we need to be worried about, and that's unfortunately where we are right now. So, I mean, I guess if I could sum up that whole time, 2003 to basically almost today, this is what most extension weed scientists have felt like in the U.S. We feel like we're leading the horse to water, but we can't make it drink. You know, I know Aaron would probably agree with me. It's just been frustrating. And about, I don't know the exact dates, but 4 or 5 years ago, I think the Weed Science Society of America started to realize we're not changing behavior with everything we've done. I mean, even though the Take Action has been fairly successful, this is a behavioral issue, and we really haven't addressed that. So what you saw start happening in weed science world is we started having a lot of symposia with behavioral economics and social economists, excuse me, just people that are generally don't come to our meetings, okay, and they would tell us, well, yeah, you have these problems and here's why, and maybe they're right, maybe they're wrong. But within my own little world, I started realizing about that same time that, you know what, this is, yeah, this is a behavioral issue, and I don't know what else I can do. But I started reading a lot of books about why farmers make the decision, or why people make the decisions they do, and that's just something that was something new to me. So something I got out of that whole time period over the past 4 or 5 years, and I'll still, you know, it's become kind of a passion for me to look into how can I do my job better by influencing that farmer's behavior. I got this from a couple of sources, and I'm not saying this is the gospel. I'm just saying here's what I think. There are some ways that we have tried to influence behavior in agriculture, whether it's companies or extension or university or whatever. One way that you influence human behavior is by manipulation. I can't do anything about this one as an extension weed scientist, as a university person, but can you think, I mean, do companies do this? Does you know, every agricultural company on the planet do this? Absolutely. Okay, so there's been attempts at this. You know, my wife loves to shop, and she loves the 2-for-1 sales, and she loves to buy one get one free kind of things, and she keeps telling me, you know, how much money we're saving, and I say, how are we saving money when we're spending it? I don't, you know, but anyway, that's just, but manipulation, I can't do nothing about that one, but it has been tried, and admirably, I'll just say, there's even Monsanto and numerous companies that have built in rewards for telling a farmer or incentivizing a farmer to apply different modes of action in their crop. Okay, they get money back, $2–3 an acre if you spray this pre-emergence herbicide, and that will help you with resistance. So you know, good efforts have been made, but not something that I can do. If you're old like me, you remember the frying pan and the egg and the commercial, and they say this is an egg, and then what do they say when they crack it and put it in the pan? This is your brain on drugs. A lot of you young people are just like, I have no idea what he's talking about. But intimidation. And so actually this is one that a lot of university weed scientists tried. I tried it. My own personal opinion is it doesn't work. But other university weed scientists think it does and have tried it and do it still today. And what I mean, well, we go into a farmer meeting, particularly in an area where there's not a problem with resistance or something. As you move further north in the U.S., you tend to see less problems with resistance. They're getting it just as bad as the rest of us, but So, so for example, some Bob Hartzler in Iowa State might ask Stanley Culpepper to come from Georgia and scare Iowa farmers to death with how bad Palmer amaranth is in Georgia. And, I don't see that as really working, at least in my own state. We tried some of the same thing. What I often heard with that is things like, we don't have Palmer. I ain't worried about that. It's a story. He's really good. He's animated and everything, but they got real issues. We ain't got it. So that's what I heard a lot out of that. There's certainly some value in it, but I don't think it's ultimately going to get us to any kind of major solving of this problem, to say the least. Another thing is association. Weed scientists were very slow to get onto this. And this is the 4 out of 5 dentist thing, and I love this one from the 50s. More doctors smoke Camels than any other cigarette. Yeah. Doctors trying to convince you to smoke their brand of cigarette. This, I say, has only been fairly new on the scene of weed science, and we have an example right here. Pat, Dr. Tranel, and Dr. Hager and Dr. Davis all have published a paper on this, and there's been some others as well. Iowa State has tried some community-based weed management. Arkansas had probably one of the first examples that I can think of where they said you go together as a area, and we call it, you sign a piece of paper, and we calling it the zero tolerance approach, and that means we have zero tolerance for any Palmer left in your field. But everybody in that area has to go along with it. You probably can't read this, but I think what in the results right here, it says, our findings indicate that high efficacy herbicide management strategies practiced at the farm scale are insufficient to slow resistant evolution in water hemp. When best practices were aggregated at large spatial scales, in other words over a community type based approach, resistance evolution was hindered. I won't read all the rest, but bottom line is, if you do things together as a community, you're probably going to come out better. If you can coerce or convince all your colleagues and neighbors that we got to have everybody on board here. Very, very difficult, I'll just say. Very, very difficult in the real world at the farmer level, at least in Missouri. Okay, where's Show Me State? Pretty stubborn guys out there, and girls sometimes, and if nothing else, the dicamba issue has shown me how very, very difficult that is to get everybody on the same page. And what do I mean by that? In many areas of our state, at least, Farmer John says, I'm going to grow Liberty Link soybeans, and by golly, I don't care if you're across the road from me, Farmer Steve. If you plant Extend soybeans and drift onto me, that's your problem, and you better not do it. So there's more of that kind of stuff happening than there is this, in my experience. It doesn't mean it can't be a success and we can't do it. We should try, but it is very difficult to get everybody on that same page, I think. The last one is what we have been tasked with in Extension. Apple is very good at this. Nike is very good at this. University Extension weed scientists, not so much. I'm not very motivational. Okay, you can probably tell already. I'm not very inspirational. I can't go into a farmer meeting and they leave there with just some heartburn saying, "Oh, I've got to do it because he just convinced me out of his heart, and that I just got to do what he says." But this is what's been put upon us, really, as university weed scientists going to talk to growers, is that you're supposed to convince them, you're supposed to motivate them to do better, and that's why we hired the I can't stand marketing as much as any person in this room, because I usually completely differ with marketing in herbicides. But that's why we hired a marketing company at the Take Action Group to give us some guidance on how to motivate people to do the right things. So all of those are kind of foo-foo, but I really kind of settled down on this, and I'm not giving up. Don't say that. I just think we need to put it in context, in that this issue is way more difficult than a lot of people give it credit for. I've got these kinds of situations in our state all the time, and that's pretty right there. That's a clean field. This guy decided to follow and do everything, I'd like to think, that we said. This guy didn't, okay? And there's a lot of reasons for that, and this is only supposed to be a 40-minute talk, and I can't go into all those reasons, but there are a lot of very good reasons for which we can't really do much about them at the university academic level, such as, hey, I rent this land and my landowner says I'm going to grow soybeans every year. That happens, okay? There's a ton of reasons why this is a very complex problem, but one of the things all the social economists did for us in this whole issue of resistance is, we know resistance is a biological problem. We got the scientists that can show all of that. We know it's a technological problem. We know there's very few mechanisms of actions or new products coming onto the market. That's the technological problem of this. Everybody wants that, but it just ain't happening right now, okay? I mean, we know this is a behavioral problem based on what I just said. If you look at the history of farmer decisions, we are very short-sighted people, okay? Short-term kind of decision-making dominates agriculture. I'm not saying by everybody, but by a lot of people. Got to make money to get through this year. You're talking to me about something, Kevin, that you're talking about something that's going to cost me more money, and I might not even see the benefit of this year. How can I do that? Those are real world things that have happened and been said to me, okay? But what the social economists said to us is, this is a wicked problem. I didn't know what that was the first time I heard it. No, didn't have any idea. This was kind of a new term introduced into weed science probably 6 or 7 years ago, and everybody started talking about, yeah, it's a wicked problem. So what the heck is a wicked problem? Problem that is difficult or probably impossible to solve because of incomplete, contradictory, or changing requirements that are often difficult to recognize. You know, hunger, food insecurity, fossil fuels. I mean, all these things are wicked problems, okay? And it may not be such a solvable problem. It's one that we need to get better at, for sure, at least in my state. I'll just say, embarrassingly, as I drive over here and cross the line into Illinois, the fields start getting a little cleaner. I don't like to say that as an extension weed scientist at the University of Missouri, but it's true, okay? I wouldn't say that in front of my Missouri clientele either, and please don't say that to them. But it's true. Y'all they're doing something different over here, and what could be different? Could be that the soil is a whole lot better, and there's a whole lot more value in managing that land appropriately compared to Missouri, and so therefore there is better management. I don't know. That's just one possibility. So, wicked problem. That brings me to the last few minutes here, and it's just, I don't claim to be able to answer this question, but here's what I thought. After everything I've said today, do you think I'm going to stand up here and say that our future is going to involve more herbicides or less? I mean, I hope you kind of got an impression what I'm talking about is we need to become less reliant, less dependent on herbicides in U.S. agriculture, because what did the guy say? Those who can't remember our past are bound to repeat it. I've had almost 16 years in one state of repeating the same thing over and over. I gave you the history and the timeline and what I've gone through with water hemp, and I can say faithfully today that the situation with water hemp is worse than when I started. I don't put that on my annual review, but so my opinion and my closing thoughts is just an opinion, is I think we're going to be dependent on you young smart people to come up with something other than just herbicides. Why do I say that? Number one, I've just said it, a seemingly never-ending cycle of herbicide resistance that we haven't really seen slow down yet. This is global increase in unique resistant case, and I should qualify that. We may have seen some moderate slowdown in the number of resistant cases if you actually track it by certain year intervals. For example, you can run the data and it will say we had one one weed increase per year during this decade, and we had a half a weed increase during this decade, or something like that. So we have done that, and there has been a slowdown in some respects, but still, that line doesn't look too good to me, okay? And so I think we are going to continue to deal with weed resistance, and I'm seeing it already with new technologies. We've had 2 years of dicamba soybeans in the state of Missouri, and we are challenging them already. Our farmers are already abusing the system and no doubt creating a dicamba resistant water hemp before too long if we keep on doing it this way. I give it 2 or 3 years at most, we'll have a dicamba resistant water hemp, because I can go to fields and I can see them spraying a water hemp that's this tall that never dies and produces seed, and they've had that field in dicamba resistant soybeans for 2 years already, and it'll probably stay in dicamba resistant soybean. That's going to create a resistant weed. We've told them this is behavioral, okay? And so number one is weed resistance. Number two, you might think this is weird, and I'm really not qualified to talk on this, but how in the world can companies even continue to put stuff on the market, I guess is my point here. And by the way, there's increasing regulatory hurdles with EPA, and I don't think that situation is going to get any better based on what's happened over the past couple of years. It takes somewhere around 11 years to bring a product to market, somewhere around $286 million, and somewhere around 250 research studies. Maybe, I mean, there's some company people sitting here, I guess, and maybe they can tell me we're willing to do that, but who's going to pay for that ultimately in the long run? It's going to be put onto the cost. They got to recoup their cost, right? It's going to be put onto the cost of the herbicide. So how can we continue ratcheting up these costs? 2005 to 2008, 256 million. 2010 to 2014, 286 million. What's it today? I mean, I'm sure it's more than 286 million. So it just doesn't seem like we're going to be able to continue at that pace without the farmers really weighing on the burden of those costs really at the farm level. And I had it in here initially, but I don't have enough time to go through it. But all our economists at the University of Missouri, the only person that I look good to follow anymore is the economist at the University of Missouri at a farm meeting, because they're not saying good things about the next 10 to 15 years of income for farming in the U.S. Okay, they make me look good when I come up there and talk to them all about all the other bad stuff. But you know, so how can we keep up at this pace? That's number two. And again, I'm not even saying I'm qualified to say that or talk about that issue, but I think it is interesting to think about. The last thing, or not the last, but one of the last is, you know, Joe Schmo on the street, or how many people in this university, if you say you're affiliated with pesticides or weed science or any of those things in any way, have a brand new great perception of you as a person. I just saw something the other day. In 2015, a survey of members of the American Association for the Advancement of Science, a survey of its members showed that 87% of those members say that GMOs are safe and healthy and no problem to eat. At the same time, a survey of the general public, what would you say that percent was? It's 33%. 33 versus 87. That's a pretty big difference, okay? And I'll just tell you, this is another subject that I'm passionate about, and I try to keep up on as much as possible, and I'll just say that number probably hasn't changed for the past decade. We keep talking about how there's a disparity and how the general public doesn't understand GMOs and pesticides. We're not doing a good enough job at educating them about it, because those numbers are very similar for the past decade or so. So that's number three. I just don't know how we're going to continue to rely on herbicides with the type of public that we have that doesn't understand it. And number four is really the purpose of this symposium and the purpose of you young people all having the youth and vigor that you do. And it's what I would leave you with is that think beyond herbicides. What else could you come up with? I've got a few possibilities here that aren't earth-shattering that everybody already knows about, but I think we need to think about things like harvest weed seed management, and this was the initial idea, and this is a picture from Stephen Powell's in Australia, and this has gone through several iterations where now it is not a tow-behind vehicle, but it's in the back of a combine as a prototype, you know, that has a weed destructor in it. And if you don't have any clue what I'm talking about, a weed destructor is just an implement, a machine. It's a roller mill. It's a machine that grinds up all the weed seed, and so the farmer can say everything going out of the back of my combine at harvest is not viable. There's no viable seed going through that combine next year. That's a great idea. That's also a thinking outside the box idea because of herbicide resistance. And it started in Australia, but it's going to happen here in the U.S. It's already here in research and in prototypes. Okay. Other than that, robotics is there's things that are going to happen in robotics. John Deere paid $300 million for a company. They don't do that just because they think that herbicides are going to be all the things we're going to do in the future. Okay. They're thinking of what our future is going to look like. Big data, robotics, GPS, GIS, all this kind of stuff. And because I told these two young people when they asked me to come speak, I said, "You realize I'm not a breeder, right? You know, I have nothing to do with breeding." There's great things that I think we can accomplish in weed science if we would just work with our breeders more to think about, can we breed allelopathic properties into our crops more? Can we make the crop be a source of weed control? There's not enough going on in that area. There's not enough weed scientists. I can tell you from a WSSA perspective, there's not enough weed scientists collaborating with these kinds of people, these breeders, and telling them how much this would be of value in the future. So I don't pretend, I don't know what this is supposed to mean, by the way, but it looked good. I don't pretend to be able to say that in our near future we're not going to be using herbicides, or even in our long-term future, but I do think we're going to have to become less dependent on herbicides, and it would be to our benefit for a lot of ways. And we're going to have to find ways to change that behavior of farmers and get them to understand that, make it economical for them to do that, and change up their tactics and not just rely on herbicides in the future. So again, I do thank you for the invitation. This is all my crew that really does the work and makes it happen all. And so with that, I guess I'll be done. And I don't know if we're taking questions or what we're doing. Yes, ma'am.
That seed destructor thing is presumably using something to recognize the weed seeds. And are you then selecting for whatever, like size or something?
Yeah.
Then are you just selecting the weed to evolve to match?
We certainly could. We could select for weeds. So I didn't explain it. I kind of tend to talk to an audience like you know what it is. But basically the whole concept is everything that runs through the combine's header, weed seed and crop, goes through the machine, and in the chaff is contained the weed seed and everything else, and that goes through a roller mill. So it doesn't matter about what size the weed seed is, it pulverizes that seed so that it's no longer viable, or at least 98% of it or whatever it is. So all of that's being worked out in research, but it's been done in Australia longer than anywhere else, and Dr. Channel might correct me if I'm wrong, but I think I've seen a paper lately that says there are starting to be species that are evolving biologically, basically, because if the weed seed is not still on the plant when the time you run through there, then it doesn't matter, right? So you're totally depending on trying to get the seed retained on the plant at the time of harvest. Lucky for us, we've done some work and other people have done some work. Water hemp and Palmer amaranth tend to retain their seed pretty well, you know, high percentage of the seed retained. It's not a complete answer by any stretch. There will be some that drops, but it could help tremendously.
So recently this summer, I just heard a very interesting idea about glyphosate-resistant weed management, and that was seeding a field with susceptible varieties. Have you heard of that, or what are your thoughts on that?
Susceptible like water hemp?
Yeah,
yeah. I don't know. Dr. Tranel's the expert in genetics of water hemp, but I don't think that would work. It's a dioecious plant, and so you're going to be sampling the genetic diversity from a large area there.
One of the things, one of the more interesting things I saw in the last 3-4 months, and you may be familiar with this quite well also, was that the similar thing to the chemical resistance to herbicides was that there have been extensive studies globally of selection for mechanical resistance, where mechanical methods are used, and the same basic phenomena such as shattering seeds or evolution to delay or to do things are identical. So that might be good to bring out for people that are not as familiar with this concept, that herbicides are not the only thing that are being defeated when you put enough pressure on them.
Yeah, they can adapt to whatever we're going to throw at them, and this example of the seed destructor, it can adapt to that over time. I'm sure it would if we selected for it long enough. It's just we need many tools, right? We need many little hammers, I guess is one way to say it.
Okay, thank you. Just a few months ago, I watched a video on YouTube, flaming technology for weeding. Do you know about that and how it works, like flame technology to kill the weeds?
Yeah, it's been investigated. No, Nebraska has done probably more than anybody I know. You know, of course, it gets into cost and feasibility of doing it across large acreages, but it is a possible method of weed control, and it dates back to the 40s. I mean, there was flaming done, but there's some new work being done with like sandblasting and stuff like that. So those are the kinds of things that I'm saying. I mean, we got to think outside the box and explore those options and make them realistic, I guess, if it can be used in agriculture. Yeah.
You know what kind of, so if we're looking to address this kind of problem, have there been modeling done that how many, for example, how many different ways do you hit something at once to make sure that this doesn't affect the problem? So if we ever get to a situation where we don't get it, is that possible, or is it we're just doomed to have increasing amounts?
I'm sorry, I can't hear all your questions.
Okay, maybe that's better. Yeah. So resistance is a very fundamental problem for everything, antibiotic resistance or any situation where you have the pressure on that things will start to evolve to kind of get around to things. Has there been any kind of modeling done or anything like this to let us know how many different ways you need to hit something to stop any kind of resistance emerging? Or are we just kind of doomed for this ultimate, like, so if you use a combination of different, because when you hear about antibiotic resistance, say, well, if you use several drugs at once, then you should prevent. I mean, it's in the same about cancer treatment, things like this. So all these kind of all have the same issue.
I'm not aware of any modeling like you're talking about, which would be good. But there has been modeling done, many different types of modeling on, just say, for example, what's the chances that I'm going to get resistance to this brand new herbicide that I put out there on the market based on things that have happened in the past and modeling. But I'm not personally aware of anything like what you're saying. Is I think what I'm getting from your question is, like, if you do X, Y, and Z, what's your chances? I mean, there's some of that, and actually Pat has done, Dr. Tranel has some examples of that where, you know, he's taken historical things that have happened in Illinois and kind of gone backwards and looked at what did they do and then what was the result of what they got. So we got we got a little bit of what you're asking for, I think, but not a whole lot of modeling, to be honest, that I'm aware of.
I must admit, like, this is not my background at all, so this might be a kind of ignorant question, but so for example, if you hit something a weed with like more than one herbicide at once, like
yeah.
herbicides, would you need to hit it with it once to stop any kind of resistance? Or, I mean, I guess that's kind of what I was thinking.
Yeah, there's some of that, and like I said, Dr. Tranel's got one of the best examples of that. It's cited and referenced in the literature all the time. It's this percent reduction in likelihood of resistance if you use 2 mechanisms of action versus 3 versus 1, all that kind of stuff. There's some of that. Yeah, I wasn't getting your question there, but that, yeah. And still a very active area of research. I don't know if you've heard this. I'm kind of just talking to Pat here, but I've heard some things coming out of Chris Preston's lab that he's saying the more we mix, some of his data is starting to show the more we mix mechanism actions, the more we might be driving towards non-target site resistance. So, any other questions? All right, well, thanks very much.
Hello and welcome to some of the new people to the symposium. Our next speaker is Dr. Elizabeth Kellogg from the Donald Danforth Plant Science Center, where she is a distinguished investigator. Her lab studies cereal crops and their weedy relatives using tools such as natural diversity and other genetic tools to sort of understand, focusing on seed production, evolution, and genomics. Her talk today is going to be on inflorescence form and function of sorghum and its relatives, so getting at plant architecture and how that can be used for the improvement of sorghum and potentially other grass species. Without further delay, let's welcome Dr. Kellogg.
I know how this works. Great. Thank you for the opportunity to be here, and thank you all for coming, particularly since I think I'm the thing that's standing between you and your lunch. So it's really been a fun symposium so far, and I think my talk is completely different from what you've already heard. But I should start out with a little preface, and that is Sir Peter Medawar, who's an immunologist who wrote widely about the practice of science, said that the scientific paper is actually a lie, because we put it together in sort of a story that is easy to follow and it fits a particular format, but it rarely follows how the discoveries were actually made. And today's talk is going to be an example. I'm going to be talking as though we had been studying sorghum all along. The experiment I'm going to talk about at the very end of the talk was actually the first one we did. But in any case, I am going to be talking, sort of hanging this story and this talk on sorghum, which I think is familiar to many of you. This is just a sorghum field here, and I'm going to be talking specifically about the inflorescence, which of course is, as you know, the seed-bearing part. And in sorghum, the flowers are born in spikelets, as they are in all grasses, and those spikelets are in pairs. So that's the first bit of morphology I want to just introduce you to or remind you of. For the purposes of this talk, I know there are a few people here who don't work on grasses. The word spikelets are the structures in which grasses bear their flowers, and for the purposes of this talk, spikelet and flower, you can think of them as being interchangeable. That's not actually quite accurate, but as I said, for this talk, you can just translate those two. So They're in pairs, excuse me, with 2 spikelets all, all the way up the inflorescence. So it's, you know, 2, 2, 2, 2, all the way up. They're kind of small, so about 5 millimeters in size. So what I'm talking about is not a large structure. More importantly, only one of these things actually makes a seed. Right? So that's the seed-bearing spikelet. And this one is usually sterile, that is, it does absolutely nothing. Sometimes it makes pollen, but it's mostly sterile, so I'll be calling it the sterile spikelet. So we've got a seed-bearing spikelet, a sterile spikelet, and the other bit of morphology that you'll need for this talk is the awn, which is an extension of one of the bracts in the seed-bearing spikelet. It's a highly modified leaf, but so those are the structures I'm going to be talking about today. The 2 spikelets, the seed-bearing one, the sterile one, and then the awn. And if you think about that for a minute, you think from the plant's point of view, this structure is stupid, right? Because it's got these flowers that are apparently doing nothing, right? They're not making seeds. And so it seems like that would reduce the productivity of the plant by 50%. And evolution should suggest select against that very easily. Furthermore, you've got this long thing sticking out of one of these that has no obvious function. So what's that doing? So these are the questions I'm going to be addressing in the talk. So what's the point of that sterile spikelet and what's the awn doing? I'll sort of answer that in a couple of different levels. One possibility is just it's an evolutionary artifact. Lots of the relatives of sorghum have this same structure, so maybe it's a holdover. Right. Possibly there's an ecological function. Maybe those things have a role in dispersal and pollination. There it is. Or maybe there's a physiological reason, and that's where I'm going to be spending most of the talk, is what the physiological reason is behind these things. And then at the very end, I'll sort of circle back to putting it into an evolutionary context with just 1 or 2 slides. So let's start with a slide that doesn't like coming up. So how do these things fall out? Where is sorghum on this phylogeny? This is a phylogeny of the grasses that was produced a little while ago by the Grass Phylogeny Working Group. And all I've done is I've taken this phylogeny and I've overlaid on it plants that or species that have spikelets in pairs, like that. And those are the blue dots. And what you can see is that there are quite a few blue dots, but they're sort of concentrated right here in this clade, which should have a box up here, there, which is the clade known by the name Andropogonae, which is the group that includes sorghum. All right, so we've got a whole clade now of things that have these spikelets in pairs, not exclusively, but this group is perhaps more familiar to you than you realize. First place, it's named for the genus Andropogon, which is the big blue stem, which is, of course, the dominant plant of the tallgrass prairie, which was the vegetation here in Illinois pre-settlement. Andro means man, pogon means beard, because they're often sort of fluffy like that. So we're looking at about 10% of the grass family. So there are 1,200 species, so it's not a tiny group. And they're all C4. I'm going to come back to the photosynthetic pathway a little bit later. What's in this group? You know the things that are in this group. There's sorghum, which I'll be talking about. Maize, which also has paired spikelets, although the pairing structure is a little different. Miscanthus, a biofuels crop. And sugarcane, biofuels again, but of course it makes sugar, which is really good stuff. And then the big prairie grasses, so big blue stem, little blue stem, Indian grass, gamma grass, are all in this particular tribe. So this is the Kanza prairie, where you can see mostly Andropogonae and then a few sort of mammalian processed Andropogonae. If you do like a Google search or something for Masai Mara or wildebeest, what you actually get is a picture of Andropogonii, which is what all this is. So the big grasslands of the world are dominated by this group of plants. If we can put specimens onto a global map where mean annual precipitation is shown up here. In the very high precipitation areas, they don't grow very well, tend to be concentrated in fairly dry areas, although not in the real deserts. So these are fairly drought tolerant critters, and mostly between 30 degrees north and 30 degrees south, with a bit of an excursion into the temperate zone here in North America. So worldwide dominant plants, ecologically and economically dominant. We've done a lot of phylogenetic work on this, been a major effort in my lab. I'm not going to talk about any of it, but I do need to acknowledge the really good people who have been doing an awful lot of this work. It's a group that's a rapid radiation. Diversification occurred in the late Miocene, so that's 8 million to 3 million years ago, which is when the grasslands expanded. They move around, as you might imagine, because of all those hairs, so they move between continents. And there's a lot of speciation caused by allopolyploidy, which I'm happy to talk about, but I'm not going into today. This work was initiated by Matt Estep, who's now at Appalachian State, Michael McCain, who's now at University of Alabama, and two really good technicians, Sami Saidi and Taylor Avishon Elder. So that was sort of a nod to the phylogenetic stuff. But I wanted to get back to the spikelet pear. Spikelet pear is actually just a short branch. It's just an abnormally truncated inflorescence branch. So in most of the grasses that, very familiar grass, things like switchgrass or dicanthelium, the spikelets are not paired. Every branch ends in a spikelet. And Andropogonii, the spikelets are in pairs, and all it is that side branch is extremely short. It ends in a spikelet. Well, it produces one side branch off of it and ends in a spikelet. And as you noticed from that phylogeny, there were things outside the Andropogonii that make spikelets in pairs. And I'm just mentioning this because it gives you a little bit of the evolutionary context. This is a paspalum, and you can see the paired spikelets right there. They form from a spikelet pair meristem, which you can see. This is a scanning electron micrograph of it. There's the spikelet pair meristem that makes the pairs. So this happens in other groups outside the Andropogonii, but the thing that's in the outgroups is that the spikelets look exactly the same. All right? They match perfectly. They're both green. They're both bisexual. They both make seeds. So unlike what I just showed you in Andropogonii, because of with these two different spikelets. Right? So the seed-bearing, sterile, and the on. The other distinctive thing about the Andropogonii is that the spikelets fall off in a different way. So in most grasses, particularly in the outgroups, the spikelets fall off individually. Okay, they just come off like that. In Andropogonii, it's actually the inflorescence breaks, and so it's the pair is the dispersal unit. So if we've got the pair as a dispersal unit, that suggests maybe that's what all this complicated morphology is for. Maybe it's just, this is a good way to get the seeds around. This is just a little cartoon. Here we've got the seed-bearing spikelet, that's what that blue oval is. And then there's the sterile spikelet, and there's the on. So that might be involved in dispersal. However, if you start looking across the whole tribe, it's not very consistent. There's sometimes the sterile spikelet stays on, as though it's helping the thing disperse. Sometimes it just falls off, and sometimes it never develops. So the dispersal role of that spikelet is not clear. It may be used for dispersal in some cases, but not in many. And for instance, if you look at, say, broom sedge bluestem, this is Andropogon virginicus, which will be blooming around here fairly soon. It's an October bloomer. What you have, here's the seed-bearing spikelet. There's a piece of the inflorescence stalk. There's the pedicel, that would lead to that sterile spikelet, but the sterile spikelet never forms. And then you've got this whopping big, straight, skinny on. So the on may be helping in dispersal, but it's not at all clear that this sterile spikelet is doing anything terribly useful. You can look at this also in things where the sterile spikelet stays on. This is the seed-bearing spikelet. That's the sterile one back here. The on coils. So the on's kind of useful. And so there are other aspects of this structure that are involved in dispersal. If we had time and the appropriate connection, I could pull up, this is a YouTube video of watching the on coil. It's hygroscopic, so that when it straightens out when it's wet, and then as it dries, it just twists. It's purely mechanical. It's not it's not using cellular energy. It just, you could put it on a lab bench and watch it crawl across the lab bench. But as it's drying out, it forces this whole unit upright in the soil. The radical emerges down here, and it produces this force downward, which shoves the whole thing obviously back upward, and that's opposed by the hairs down here. So the whole thing forms a kind of a useful dispersal unit, but it's not at all clear that this thing is doing anything much about that at all. So kind of ambiguous data in terms of the role for this in dispersal. And if we get back to sorghum, in wild sorghum, things like Johnson grass, which is a sorghum species, that sterile spikelet just falls off before dispersal. So this is the fertile spikelet. These would have been the sterile ones, and that falls off. And the on is kind of wimpy, so that the on is not. I haven't done the experiments to prove this, but I'm not sure that the on can generate enough force to force this thing up into the soil, because it's pretty heavy. So, and then of course, if we're talking about cultivated sorghum, none of that's relevant at all, right? It's dispersed by humans. So, the sterile spikelet is present in cultivated sorghum. On is sometimes there and sometimes not. Well, what about pollination? If you start looking through the literature on sterile flowers, what's the good of sterile flowers? What you get is things like this viburnum, where it's got, these are sterile flowers out here, and these are seed-bearing flowers in the middle, and people have done the experiments to show that indeed if you take off these sterile flowers, it reduces seed set in the fertile ones because you've lost the pollinator attraction. That's great. It's definitely well documented, but of course grasses are wind pollinated, so that's irrelevant. Right? So it's obviously they're not helping attract pollinators. Sometimes, and in some wild Andropogani, that sterile spikelet is actually staminate. So it can be affecting pollen-olive ratios. You can see that this is big blue stem early in development. This is the seed-bearing spikelet over there on the right. Let's see this pointer. There's the gynecium, that'll be the seed, and it's 3 anthers. This is the sterile, in this case, pollen-bearing spikelet, 3 anthers, and that gynecium is going to abort. So it's a very active process. Seems to be the same process, same cellular genetic process is involved in making male flowers in the tassel in corn. So certainly possible involvement in pollen production. It might correlate with the breeding system, but in fact for most grasses and most Andropogani, we don't actually know what the breeding system is. There have been almost no experiments. I think there are only about 15 or 20 species of grasses in which we actually know whether they are inbreeding or outcrossing, which astonishes me. So if anybody wants something to do, you could answer that question. Maybe having extra pollen will increase the likelihood of fertilization when it's there, but we don't actually know if any of these things are pollen limited. So once again, some very basic biology that is simply unknown. So we don't really know. But certainly getting back to sorghum, in most sorghum lines, that sterile spikelet is truly sterile. There are no floral parts in there. I've checked. So it suggests not only are these sterile spikelets not involved in dispersal, but they're probably not involved in a lot of pollination either, at least in this particular crop. So, right, so what are these things doing? Well, and this is where we get into physiology and these things that you look at for many years and don't pay any attention to. That the sterile spikelets are green. And at some point, I got thinking, well, wait a minute. If they're green, they're probably photosynthesizing. And if they're photosynthesizing, where's the carbon going? The other point is the ons. The ons are obviously kind of brown, not a lot there. But if you look at the literature on ons, there are some well-documented studies starting in the 1950s and 40s and 50s and summarized here in this nice review from 1963 and other more recent papers saying that when you look at wheat, so this happens to be reporting on durum wheat, used carbon-14 to trace the carbon, appreciable amounts of photosynthate were translocated from the ons to the developing seed. And so if you look for the function of ons in the literature, they'll cite the wheat studies. There have been similar ones in barley and similar ones in rice, saying, well, ons are actually feeding the seed. So now we've got a couple of possibilities. One is that the ons in sorghum, even though they don't look awfully green, might be feeding the seed. And this pedicellate spikelet, or the sterile spikelet, must be doing something. So the question is, do these things provide carbon for the developing seed? And we went after this in a bunch of different ways, and I should say at this point, this has been the most evenly balanced collaboration I've ever been involved in my life. It took us a while to figure out how we were going to structure the authorship of the paper, because we didn't think we could get a journal that would accept us with 5 co-first authors. But Doug Allen is a colleague of mine at the Danforth Center and also at ARS. He's a biochemist. This would not have been possible without him. Taylor Obashan Elder was involved in virtually all the experiments. Victoria Koneva also involved in pretty much everything. And David did a lot of the bioinformatics at various points. So really a team effort. And what we did was we started looking at carbon-14 labeling, because that was what was in the literature. We then moved on to carbon-13 using LCMS-MS, RNA-seq, spikelet removal experiments, and also just a little bit of carbon-11. So I'm just going to walk through some of those experiments and what the results are. So starting with carbon-14, we did pulse chase experiments. So we took an inflorescence and either exposed it to carbon-14 as a whole, or just to test what the parts would do themselves, we dissected it into the sterile spikelets, the fertile spikelets, and the ons, and just had them just sitting there at the base of the flask. So either way, so we've got intact or detached parts exposed to carbon-14 for an hour, and then we just asked how much carbon-14 was taken up by each individual part. We then did a chase experiment where we did that same 1-hour experiment, then replaced the carbon-14 with fresh air, and let the thing just sit there and photosynthesize for 24 hours. And so that second bit would tell you not only what gets taken up, but where it goes, because you're not having any more label to take up. So just to walk you through these results, after an hour of pulse, the on basically did nothing. So it's less than 1% of the counts, the radioactive counts on the counter. The sterile spikelet took up most of the largest percentage of the carbon that was in this structure, was almost 90%. The seed-producing spikelet, about 10%. So it wasn't zero, but it wasn't anything to write home about. The interesting part, of course, came after 24 hours. A little bit of the carbon ended up in the on, which meant a corresponding decrease in the sterile spikelet, and much more in the seed-producing spikelet. So this suggests then that the on is doing nothing, but that the sterile spikelet here is functioning kind of the way the on does in wheat, as kind of provisioning the tissue. This is just the same data shown as a bar graph, where the sterile spikelet is shown in orange, the percent of carbon-14 counts, decays per minute per million. And you can see it, whether detached or attached, intact after 1 hour pulse. It was a lot in the sterile spikelet, but then it gets reapportioned after the 24-hour chase. So the purple is the seed-bearing spikelet, and then the on is just that little teeny orange bit down there. Now, I mentioned at the beginning this is a representative of the whole tribe. So we looked at a couple of other species in this same tribe. That is this just a sorghum thing, or do all the plants that have this structure go in for the same thing? So we looked at Themata triandra, which is a major range grass in Australia and South Africa, and you get basically the same answer. That is major uptake by the sterile spikelet in 1 hour, and then in chase, the carbon appears to end up in the seed-bearing spikelet. And Andropogon chirensis, another major range grass from southern Africa. Same pattern. Okay, and these are unrelated, so they're within that tribe, but they're in different clades. So it seems to be general. We looked at the surface, just the epidermal surface of these things, just to say, well, is there any way for this carbon to get in? And if you look at the sterile spikelet, it looks like a leaf. So you've got the lines of stomata here, right here. These are just silica bodies and prickle hairs. This is a very leaf-like sort of thing. So plenty of stomates around. If you look at the seed-bearing spikelet, lots of silica, but basically almost no stomata. Taylor decided she really needed to check more thoroughly than I had done. And indeed, when she looked right up at the very top corner of the sterile spikelet, or the seed-bearing spikelet, where it's just very slightly green, there are indeed a bunch of stomates, but this is in a very small region. So it's again, it's back to the idea this is taking up some carbon, but not a lot, not as much as the sterile spikelet. And if you look at the on, it's pretty, but there are no stomates on that guy at all. And this is true for the other couple of species as well. So at that point, Doug, being a good biochemist, got curious about exactly where this carbon was going. Just what compounds was it ending up in. So we did a very similar set of experiments, but labeling with carbon-13, which allows us to check then metabolites on the MS. So expose the entire inflorescence to carbon-13 for 30 seconds, 60 seconds, and 5 minutes, just to see where it was going. Then freeze the thing and dissect it into the same 2 spikelet types in the ons, and assess the labeling with the mass spec. And we focused specifically on metabolites that would be involved in photosynthesis and in glycolysis and starch metabolism. So we weren't just trying to get everything. It was really the question of, are these things photosynthesizing? Are they breaking down carbohydrates? And I'm not going to read this entire ANOVA table, but the results were really pretty consistent. So that if you look across like what this table is, it's listing the metabolites across the top. The primary ones for C4 are aspartate and malate, and then the other ones are involved in other aspects of metabolism, and then the various comparisons down the side. So if you compare the seed-bearing spikelet with the on, what you see, so that's that comparison, it's entirely non-significant. Okay, they're very, very similar in terms of the metabolic profile. But if you compare either of those 2 structures with the sterile spikelet, the substantial and significant differences in all the metabolites. That is, the sterile spikelet is clearly doing something quite different from the other 2. And in terms of the time courses, labeling time, no difference in the first minute, but by the time we've let the things label for 5 minutes, that's significant differences there. So what you're seeing is that by 5 minutes, there's a substantial difference in the metabolite profile of the sterile spikelet. You can just see that this is a principal components analysis of the same data. So this is all these all these symbols represent the on and the seed-bearing spikelet. They're not significantly different, and they're all piled up at one corner of the PCA, whereas the sterile spikelet by 5 minutes is metabolically quite distinct. And if we, and I should say we did the same thing with Themata triandra, and you get a very similar pattern. So once again, it's repeatable across species. It's not a sorghum thing. So just to put this on a cartoon of a cell, you've got carbon dioxide coming in, and then the first number in each set of brackets is the percentage of the label that's in that compound. So in each, in the sterile spikelet, the second number is the seed-bearing spikelet, and the third number is the on. Okay? And what you can see just looking across this cartoon is that the numbers for the sterile spikelet are relatively high, and the others are fairly low, close to the background on the mass spec. So just to summarize, it's the seed-bearing spikelets are appearing to import carbon from the sterile spikelet. The sterile spikelets are photosynthetically active. The ons are not metabolically active, either for carbon uptake or breakdown. So we have no idea what those ons are doing. And Themata is similar in all these experiments, was repeated there. But we got an unexpected result, and this is where I need to come back to the C4 story. Sorghum is C4. And so the first products of carbon fixation should be 4-carbon compounds. And it does happen that way in the sterile spikelet, but the numbers are really not as high as expected. So in fact, this level of incorporation in malate and aspartate, which should be the first thing, is not as high as we thought it should be. And that seemed a little weird. So we weren't really trying to establish the photosynthetic pathway in this particular spikelet, but since we had the data, we started looking. And before I go a whole lot further on this, I think I need to just remind you of photosynthesis so I can mention some enzyme names without everybody going to sleep. So let me just remind you, this is just intro bio, of course. Carbon dioxide going in through the stomates. And if you do a cross-section through the leaf, here's the stoma here, so the carbon dioxide goes in there and ends up in the mesophyll, gets taken out of the intercellular spaces in the mesophyll, and ends up in the chloroplasts, like that. And of course, all photosynthesis, almost all photosynthesis on Earth goes through this enzyme. This is ribisco, ribulose 1,5-bisphosphate carboxylase, and the ordinary C3 pathway starts with a 5-carbon sugar. Carbon dioxide binds to that. You get an unstable 6-carbon compound, which falls apart into 2 3-carbon molecules. I'm sorry, for those of you who like the hydrogens and hydroxyls, I left them off. I apologize. Just trying to remind you where things are. And if you label it, label it with something like carbon-13, like what we've been doing. The first earliest product of photosynthesis in an ordinary C3 photosynthesis is a 3-carbon compound. That's why it's called C3. Right? It's the first thing you can detect because you can't see this 6-carbon thing. Right? This is the picture from the textbook with all the other pieces on it. So it's just the same thing I just showed you, but it's got everything that's hanging off the carbon. So 5-carbon ribisco, giving you 2 molecules of 3-phosphoglycerate. All right. C4, of course, you start with a 3-carbon molecule. It's just phosphoenol pyruvate. Tack the carbon dioxide onto that, and that gives you a 4-carbon molecule, oxaloacetate. That's why it's called C4. Okay. And then that gets reduced to malate, or there's an amino group put on, you get aspartate. Okay. So malate and aspartate are pretty early. And the enzyme involved here is PEP carboxylase. So part of our problem in detecting these metabolites was that malate and aspartate turn up in lots of other cellular functions, in addition to just this particular bit of photosynthetic metabolism. And that's true for a lot of these other compounds as well. I mean, for instance, if you think about pyruvate or the triose phosphates, if you remember glycolysis in the Krebs cycle, you remember it, right? There's the bit that comes down and the bit that goes around. Yeah, and pyruvate's right where the bit that comes down meets up to the bit that goes around. Yeah, and the triose phosphates are just a little above that. Okay, anyway, so that you find these things and we label them. We haven't differentiated explicitly with all these compounds. Some of the compounds are specific, but with many of them, they're not specific. So one possible solution, of course, is to check the gene expression of the genes that are producing these things in the different pathways. So if Rubisco or the C4 copy of PEP carboxylase are highly expressed, then the sterile spikelet is probably photosynthetic. So we complemented the metabolite work with RNA-seq, just testing whether they were reflecting the biological processes we thought they were, and basically looked at our particular set of metabolites and then looked at the genes encoding the enzymes that would be producing those. And we compared that to a leaf, which we know is a perfectly good C4 thing. So this is why it took a while to explain this, because this is very hard to read. So this is a heat map showing gene expression in the leaf, the sterile, or the seed-bearing spikelet, that's SS, the on, and the sterile spikelet over here. And the colors are really washed out on this slide. So you may have to just trust me a little bit on this. And then each line across represents expression of a particular gene. It's all relative. Okay, so it's just all normalized, so it's to between 1 and -1. So if it's high, it's given a 1, and if it's low, it's given, if it's high, it's dark red, and if it's low, it's blue. So what you can see immediately from this is that the leaf has a whole bunch of genes that are very highly expressed, a whole bunch that are basically off, and the seed-bearing spikelet is the opposite. So the seed-bearing spikelet doesn't look like a leaf at all. The sterile spikelet, somewhat to our surprise, so it's this column here, was kind of intermediate. That is, it doesn't look exactly like a leaf. It's not really cranked up all the big photosynthetic enzymes, but it's not cranking up all the starch synthesis enzymes either, or the glycolysis, all the heterotrophic metabolism enzymes that you get in the seed-bearing spikelet. And the on is just peculiar. Just zooming in here just on a little bit, this is just the photosynthetic enzymes. This particular row is the C4 isoform of PEP carboxylase, which you can see is very strongly expressed in the leaf, basically off in the on and the seed-bearing spikelet, and it's there but kind of low in the sterile spikelet. As though the sterile spikelet might be photosynthesizing, but it's not cranking the way the leaf is. So this is consistent with our metabolite results. So here's another C4 specific enzyme, and you've got, pretty much the same pattern. So that got us thinking, well, all right, what we've got is a tissue that seems to be photosynthesizing, but it certainly isn't leaf-like. What about the anatomical structure? And again, a reminder about C4, to make C4 work, the cell that's capturing the carbon, the mesophyll cell, has to be right next to the cell that's reducing the carbon, because the C4 requires things to shuttle between the cells. And that means that the veins in C4 plants are closer together than in C3, so that if you look at, for instance, here's a chloroplast in a mesophyll cell, here's a bundle sheath chloroplast, carbon dioxide comes in, turned to bicarbonate, and added to a 3-carbon compound. That's the 4-carbon compound for C4. That 4-carbon compound gets shot into the bundle sheath, and then that CO2 gets taken off and handed back to Rubisco. It leaves a 3-carbon compound, which then that 3-carbon thing has to get shuttled back out here to the other one. So this is basically the C4 cycle. It's a 2-cell pathway, and you've got to be able to move these things back and forth. That means if you look at a C3 versus a C4 plant, you can just tell the difference based on the vein space. All right, so here's a C3 plant. This is Dicanthelium oligoxanthes. This was some sections done by an REU student in my lab, Jeffrey Yen. You can see there's a vein here, and then there's a whole bunch of cells, and then there's another vein, and then there's a whole bunch of cells, and there's another vein. They could be a long way apart. On the bottom is maze, which is C4, of course. Here's a vein, a couple of cells, a vein, cell, vein, cell, vein, so forth. Things are just jammed right together. So the question then is, what does the sterile spikelet look like? And there it is, and there what you can see is the vein spacing, where they're not as far apart as in a C3, but they're certainly not as close together as they are in a sorghum leaf. Okay. So all that suggests, both the metabolite and the RNA-seq data, suggests that the sterile spikelet is certainly doing more photosynthesis and more C4-like than the seed-bearing spikelet or the on, but less than the leaf. Okay. So, right. So everything I've been talking about so far has been biochemical and anatomical. And you might reasonably say, well, what does this have to do with the whole plant out in the field? So the experiment we then tried, we being Taylor, was to just say, well, what happens if you take off the sterile spikelets? That, you know, if they're so good, then what's the deal? So since there's a lot of diversity in sorghum panicle architecture, and we weren't sure which one to work on, we just chose 8, just because they grew well. Did 8 different lines of sorghum, different structures, and removed the sterile spikelets from alternating inflorescence branches. And so what she did was, she had, if you've got this big dense panicle, she took the sterile spikelets off of this branch and left them on that one, then took them off this one and left them on that one, and so forth all the way up. So it integrated all the way up the panicle, and then let the thing grow to maturity. It's a very conceptually simple experiment. It turns out it's incredibly labor intensive. So we collected those and compared the average seed weight between the branches with the spikelets removed and the ones with on, and we did several other controls that I'm not going to march through. And we did that the same with 5 of these lines had ons, and so we took the ons off as well. So what happened? Well, if you remove the sterile spikelet, it reduces the seed weight. Each one of these pairs of boxes is a separate accession, so these blurry letters across here are the accessions, and the ones with the spikelets on, staying on the plant, are in yellow, and the ones with them removed are in pink. And there are a couple of exceptions, but in general, for those of you who like BTX 623, which is the reference line, that's the one all the way over at the right. It's very clearly different. In general, the trend of the direction of the trend is pretty much the same. As you take those pedicellate spikelets off, the sterile spikelets off, and it reduces yield, reduces seed weight. Some of these are significant, some of them are not. If you combine all of those into just a single linear model and say what's the effect of spikelet removal, you get this, which is about a 5% reduction, statistically significant reduction. 5% does not look like or seem like very much. However, the value of the sorghum crop in Missouri last year was about $20 million. So these sterile spikelets, all 4 millimeters of them, would be worth about $1 million in terms of contribution to that grain yield. Removing the ons does basically nothing. No significant effect. So when we ask what's the function of the sterile spikelet, sometimes it produces pollen, sometimes it's involved in dispersal. It may always be involved in grain filling. And the function in cultivated sorghum seems to be grain filling, where it's providing a nurse tissue that contributes to yield. Now, when I've given this talk before, I usually get to that point and somebody says, well, why doesn't the plant just put a whole leaf in the inflorescence? Right. I mean, we're back to the plant being stupid again. And so that's a reasonable question. And in fact, there are many of these Andropogoni that do that. So here's a phylogeny Michael McCain produced a while ago. as just an ordinary phylogeny, but in the clades that I've marked in blue, they actually do put a leaf in the inflorescence. There's a bract under in the inflorescence branch, right next to it. And fortunately, one of those was one that we had already, we've talked about sorghum. One of those is ones we've already sampled, and that is this range grass themata. Okay. So this is what that one actually looks like. It's a pretty complicated structure, but you can see the awns that I've talked about before. The sterile spikelets surround the seed-bearing spikelet, so you can't really see them here. But then this is a leaf-like bract, which is separated from the sterile spikelets by an internode of like 2 or 3 millimeters. So it's very close. And we did the carbon-14 labeling on this. And after 1 hour pulse, the awn didn't do anything. Seed-producing spikelet was 3% of the counts. The sterile spikelet was, when we look at the whole thing, including the bract, was 28%, and the bract was 69%. So the relative proportions of sterile seed-producing and awn are the same as what I've already showed you, the same as what you saw in sorghum. It's just when you add in the bract, then that becomes a smaller fraction of the whole. And then after the chase, some of the counts end up in the awn, a bunch in the sterile spikelet, and a bunch in the seed-producing spikelet, and there's a big drop in the bract. So having a bract in the inflorescence is actually a good deal. And so here are the bar graphs of that same thing. The bract, whether detached or attached, takes up carbon, but then that gets reallocated among the other spikelets after the 24-hour chase. And this is the metabolic data. This is just the repeat of what I showed you for sorghum, where the awn and the seed-bearing spikelet are not significantly different. The sterile spikelet is different 5 minutes. But when you now add the bract data into that, this whole graph sort of gets relatively compressed, because this is just a relative percentage of counts, or relative percentage of metabolites, sorry. So what I just showed you was that bit there, and then the bract, the bract is distinctly different, particularly after 5 minutes. But so the bract is clearly metabolizing in a different way from any of the spikelets structures. The other thing that we could do, and as I said at the beginning, this was actually the first experiment we did. Other thing that we can do is actually label this with carbon-11, which where you can look at it in real time. And I have a movie, but I don't have it embedded in this particular talk, so you'll have to believe me when I tell you what the movie shows. But this bract is big enough that you can actually put that inside a chamber by itself, feed that chamber with carbon-11, and then watch where the radiation signal goes. And so this was a collaboration with some folks at the WashU School of Medicine in the Department of Radiology. So this is basically a PET scan. Okay. And so we add the carbon-11 there, and the pictures are a little hard to see, what I'm going to show you in the next slide, because this bract gets so hot. There's just so much radiation. Carbon-11 is a very strong emitter, gamma emitter, that's primarily what you see. But you start seeing other things as well. So here was this, the other thing is this particular picture was taken with a cell phone. So this is the structure that you just saw taped down onto a board. And then there's the bract all lit up. And after about 3 hours of labeling, you see label coming up in the spikelets as well as going down into the node below. So it's a bidirectional transport of carbon from this photosynthetic bract. 3 hours is about as long as you can do the experiment, because you're running into the problem of the decay of the isotope. So what do we know? The sterile spikelet is producing photosynthate, some of which goes to the seed-producing spikelet. The awn is not contributing to photosynthetic metabolism. It actually does have some interesting gene expression, but it's clearly not photosynthetic. Having a leaf-like bract in the inflorescence makes more photosynthetic more photosynthate than just the sterile spikelet, which still leaves open this question of, well, if it's such a good deal, why doesn't everybody have a bract in the inflorescence? And, I don't honestly even have a good hypothesis to test with that. So if anybody's got any bright ideas, I'd appreciate hearing them. And finally, just back to the evolutionary story, I showed you this at the beginning, that this photosynthetic function and seed-producing function of the spikelets is likely ancestral. So remember these guys, this is the ancestral condition for the paired spikelets. They're both green, they both make seeds. And so it appears that a novel characteristic in the Andropogonii is that the seed-bearing spikelet has retained its ancestral seed-bearing function, but has lost its photosynthetic function. And conversely, the sterile spikelet has retained its ancestral photosynthetic function and has lost its seed-bearing function. So that the sterile spikelet is now a nurse tissue rather than a seed-producing tissue. So I just need to, again, acknowledge the phylogeny team, which is quite large. Most of them have moved on to other things, but a lot of people have been involved in phylogenetics. And then most of the work I told you about today was in close collaboration with Doug Allen. It would not have been possible without him. Victoria Koneva, Taylor Elder, and I guess I didn't get David Goad's name up here. But anyway, that's the team, and just I can take some questions while you look at some pretty Andropogonii. Great. Thank you.
Do we have any questions?
We have lunch.
Just the other side of the room.
Yeah.
I'll wait for the mic.
Fantastic talk. So what type of photosynthesis does the bract do?
I assume it's C4, but it looks that way. I mean, the bract looks very like when you do the in the RNA-seq data, it looks pretty much like the leaf. Yeah.
How critical do you think the benefits from having a bract was? Like, is it contributing a lot of?
A lot. So you mean in terms of the carbon economy of the whole plant?
Yes.
Yeah. We don't know. We don't. That would be possible. These are long-lived, very large perennial plants. So they're in the wild they're like this tall, big. So it would be a challenging experiment to do, but probably not impossible. We have one thought about all of this is that a lot of these things grow in areas where there's seasonal drought and also fire. And we're kind of wondering if say, the bract or even the sterile spikelet might be some sort of a backup kind of thing or insurance thing that if you get hit by drought or a fire at a particular stage in the growth pattern, does that kind of rescue the inflorescence? Or so that you're not dependent, you might not be dependent as heavily on carbohydrate coming from the leaves. That's a complete hand-waving story, except that it's a little bit consistent with some of the data on getting back to wheat and barley and the awns, where the amount of that those contribute to the grain economy depends on the moisture level. So particularly in drought years, they're more important.
So I guess it could be possible that since there's since this bract and sterile photosynthesis from bract and sterile spikelets are backups, it might be possible that the difference that some grass might be just like accidental, like evolutionary accidents.
It could be an accident. Yeah, that's always the one option. But yeah, I mean the challenge with these hypotheses is trying to figure out some way to actually test them as opposed to make them a plausible story.
I think your results about the C4 and the spikelet were really interesting, particularly with the fact that, as you showed, it doesn't really have crowns enough.
Yeah, not very good.
Yeah, so this is kind of interesting. I'm thinking of things like the C4 Rice project where they've been trying to introduce this. And one of the major problems has been getting any kind of unitype without hands and atoms, which obviously you can't currently engineer. So I guess kind of continuing on from Anthony's question, so if you think C4 may be happening, what's kind of happening to further respiration, I guess, in spiders? And then also, has anything been done looking at actually where the enzymes are deficient? Has it been like kind of in situ to show that or anything like that?
No, there hasn't been anything. I think to my knowledge, this is the first data on any of this actually. I don't know anywhere in the literature where any of this function has been explored or even suggested. We did check the labeling of glyoxylate, phosphoglycolate, and there is a little bit of low labeling. The caveat with that is it doesn't label very well generally. Even when you've got the photorespiration going on, it's a harder to track. So there's a hint that there's some photorespiration, and hence perhaps again it's sort of a mushy C4. It may be some C3, some sort of mixed. I'm hesitating to use the word intermediate. I'm guessing it's probably more mixed, but yeah, you need to do the in situ real localization. Sorry, what?
I guess it'd be kind of cool to see a slight improvement, like surface, like a C3 type anatomy, just so that some kind of slight improvement.
Yeah.
Optimistic.
Yeah, no, the vein spacing is not as far apart as I would have expected for C3. But yeah, they're not supported.
So, can you hear me?
Yes.
What, in terms of grain filling period disorder, how long does that period last, and is the sterile spikelet functioning to feed carbon during that whole period of time, or is it something that's just developmentally limited?
So I don't know the answer to how long the grain filling period is. We did all these experiments in anthesis because I was concerned that having a very strong endosperm sink would confuse the issue. But yeah, one of the next steps clearly would be to do this, maybe just the carbon 14, because that's the simplest of the experiments. Do that at various different times. There's some point at which the sterile spikelets dry up and fall off. So I suspect this is a transient thing, and the only reason we know that it matters a lot is the removal experiments. So that there's some stage where it's still contributing enough, even if it doesn't do it for very long, but it's going to make some difference.
So in the removal experiment, I'm assuming that 4% or 5% change represents the amount of photosynthate that's coming from the sterile spikelet versus what's coming up to the stem from the rest of the plant.
We would assume that the flyleaf is, all the data I know about is that the flyleaf does most of the work in terms of grain filling. But it's a long way away.
Hello, I just had a quick question about your sterile spikelet removal experiment. So you did alternating branches, right? And you said you saw a decrease in seed weight. And did you see it, did you notice if any of the viable spikelets, there was any sort of loss, organ loss, like abortion or something that occurred when you removed the sterile spikelet, maybe kind of highlighting its role in your seed organ?
Not that we saw, but the experiment wasn't done in a way to be able to detect that. We did have control plants where we had just a whole separate plant in the same pot that wasn't touched. And that was indistinguishable from the branches that were not manipulated. So there's some suggestion that, the manipulation per se wasn't screwing up the whole plant. I don't know if that gets through.
Well, just seed number, I guess.
Yeah, yeah. No, it was average. You know, it was the average weight. Yeah.
Thank you so much. Do you think there's like a big number of density of stomata in sterile spikelets as compared to the seed bearings, but is there a number of stomata difference?
Yeah, yeah. So the sterile spikelets have they look very leaf-like. They have, you know, sort of the stomatal density that well, we didn't measure. So I can't say for sure it's exactly the same as leaves, but it's what you would expect if you looked at a leaf. Whereas the sterile spikelets, they were hard to find. You could find a few, but in general, and that was true for all tree species. Actually, on Pemmica, we couldn't find any at all on the seed-bearing spike. Seed-bearing spike, and they tend to be very hard and shiny, so the whole structure of the bloom is really quite different.
Do you have any other questions?
So towards the end, you propose that there's a separation of function, photosynthetic function, and seed filling function. So, was the plant successful in that strategy? In other words, is the sterile spikelet capable of producing photosynthate at a rate that's twice the rate of a single ancestral fertile crop?
Yeah, it makes sense, and it's a great question. We don't have those data. Yeah, but it would have to, something like that would have to happen, because obviously otherwise it would be a net loss.
If there are no other questions, I'll just scan the room. No. We have lunch in the heritage room upstairs. Please come, take lunch, take 2, take 3. I shall count attendance. Take 1. 1, 2,